Okay, so we're going to start with a little bit of a review of aerobic respiration before we go on to what's happening in anaerobic respiration. So we have already talked about our starting molecule of glucose. This is it. Our glucose C6H12O6, and it is in the process of glycolysis. Broken down into pyruvic acid or pyruvate, and it's broken down into two of those. That pyruvate then heads to the Krebs cycle, where the end result of our Krebs cycle is the making of quite a few electron carriers in the form of NADH and FADH2. These electron carriers then head to the, let's put my box in here next, the electron transport chain, which results in production of copious amounts of ATP. Right? So we can fill in a few things along the way here. One of those is over here in glycolysis, we are going to use a little bit of ATP, 2 to be exact, and we're also going to have made some, we're going to use ATP, that's going to become ADP, my apologies. We're also going to take and make 4 ATP. And these things, this ATP here is going to be used by the cell. So it's going to regenerate its ADP after it's used, and there's constantly a source of ADP in cells as ATP is constantly being used and regenerated. Now, glycolysis is also going to be responsible for making NADH from NAD positive. So our NAD needs to be reduced in order to make our final pyruvic acid here. So these NADH that are made are going to make their way all the way down to the electron transport chain. And in the electron transport chain, you can see that video where it talks more specifically about how this happens. But the electron transport chain is going to strip the electrons from our electron carriers, and we're going to be left with our emptied, our oxidized, electron carriers of NAD positive and FAD positive. These guys are going to go back to the Krebs cycle where they get re-energized, or in one case, they're going to go all the way back up here to glycolysis where it can be used in that breakdown of pyruvic acid. Right? In order for the electron transport chain to happen, just a reminder here that oxygen is constantly being used as an electron acceptor. So as the electrons are dropped off and utilized to make ATP, those electrons get picked up by oxygen and hydrogen and make water as a waste. Right? So this oxygen is fairly important, and now you know why you breathe in and out every day. If you don't have oxygen, then there's nowhere for the electrons to go. If there's nowhere for these electrons to go, then you can't regenerate your electron carriers. And if you can't regenerate your electron carriers, then the Krebs cycle fails, glycolysis fails, and you have no more production of ATP. So this oxygen is very critical to the flow of cellular respiration as it picks up those electrons. So why do you breathe in and out every single day of every moment of your life, whether you're asleep or awake? And it is because this oxygen is so necessary for the process of cellular respiration. All right, that's a little aside. So this part here, from here down, along with glycolysis, this is where we're focusing on our aerobic. And we can extend aerobic, although oxygen isn't used up here, these go together. So aerobic respiration is made of the Krebs cycle and electron transport chain following glycolysis. Now, what happens if there is no oxygen? What happens if the organism in question, or an organism doesn't have mitochondria, or they're short on oxygen? in order for glycolysis to continue, we still need this NADH to be regenerated. And in aerobic respiration, that's happening because it goes down to the electron transport chain. But this is a barrier, this is a stop. You need O2 to go past this point. You need O2 to go past this point. So if there's no oxygen or no mitochondria for these cycles to continue, then our pyruvic acid is left over and needs to be dealt with. And those pathways to deal with the pyruvic acid are called fermentation. And we're going to talk about two of them just briefly. These are overviews. We're not going to go through all the molecular changes that happen in fermentation, but we will say that fermentation's purpose is to regenerate NAD positive, our electron carrier. We are going to tell us that there is no ATP, pardon the bell, no ATP made. And on top of that, this happens in the cytoplasm, which happens to be one of the parts of cells that every cell has, whether they're eukaryotes or prokaryotes, etc. All right, so the two different types of fermentation then are going to be one that results in the making of lactic acid, and this is called lactic acid fermentation. The process of making the lactic acid from our pyruvic acid, so our pyruvic acid is coming out here to the cytoplasm, well, it's already in the cytoplasm, and the process of breaking down and making that lactic acid is going to release our electron carrier NADH to go back to the breakdown of glucose for another round of glycolysis. The other pathway of fermentation we're going to mention is that of alcoholic fermentation. And alcoholic fermentation results in the production of ethanol and carbon dioxide. And this pathway is also going to take NADH and it's going to recycle it into the open electron carrier, which again can cycle back up here to glycolysis. So this then is our anaerobic pathways. Those are anaerobic, right? Glycolysis in combination with fermentation. A couple things to be said about our two different types of fermentation here. Lactic acid fermentation is what would be happening in some animal cells. This is what is going to happen in some of your cells where the demand for oxygen is so high because you're putting a lot of effort and work into those muscles, so that those cells are still going through cellular respiration on an aerobic level, but they can't quite keep up, and you're having more pyruvic acid made. Then that pyruvic acid, to regenerate the NAD positive so glycolysis can continue, that pyruvic acid can be broken down into lactic acid. So animal cells, some types of yeast or fungi are in here, and lactic acid is reusable. So lactic acid then can be turned back into pyruvate by many organisms. So this isn't a dead end. You can go on and keep using that. The ethanol then and carbon dioxide that is used here is the ethanol and CO2 is a part of a dead end process. So alcoholic fermentation, which is this lower one, is a dead end process for a couple reasons. The first is that ethanol can't be reused by that cell and broken down. And the second reason is that ethanol, as it builds up and increases in concentration, is toxic to living things. If you think about what we use alcohol for besides cooking or drinking, we might find it in our bathroom as rubbing alcohol. It's used to treat wounds, to keep things from getting infected, and to clean materials before we use them. So alcohol itself is a toxin and poisonous. Thus, if you're a small cell and you are going through alcoholic fermentation, this ethanol is a dead end, one, because you can't reuse it, and number two, because as it accumulates and the concentration rises, you will end up dying from the toxicity of your own waste. Lactic acid, on the other hand, is not a dead end process, because at the end of this fermentation process, this lactic acid has the potential to be regenerated or recycled into pyruvate. And then the pyruvate could go all the way back here and enter the Krebs cycle. And this is something that humans do, and we see this in our muscles when we do a lot of intense muscle work for a period of time without a break. So running, we often see this in quads and calves. If you're lifting weights and you're doing a number of repetitions very quickly, you may be taxing your muscles, and they may turn to some lactic acid fermentation. And that lactic acid, once it's removed from your muscles, can be regenerated into pyruvate and used by the Krebs cycle. And there's a lot more to that, of course, but generally speaking, it can be reused.